SYTOX Deep Red is a counterstain with impressive applications for revealing higher-order chromosome organization, a topic pertaining to the development of cancer and many other diseases. Therefore, I tested both a 1 to 4,000 dilution and the manufacturer's 1 to 2,000 dilution of SYTOX and compared the results to other commonly used counterstains, such as DAPI and topro-3 iodide. Metaphase spreads were used to test the fluorescence of SYTOX Deep Red. The immortalized retinal pigment epithelial 1 cell line was stained after bursting its cell's membranes. Through confocal microscopy, fluorescent images were analyzed and revealed the 1 to 4,000 dilution to be more resourceful than the manufacturer's dilution of 1 to 2,000, both of which were notably better than the traditional DAPI counters. While the metaphase spreads did not produce any mitotic cells, this can be explained by either a 2-confluent cell plate, which would have activated density-dependent inhibition of cell division, or it can also be explained by the absence of the chemical Coopersmith, which is usually used in metaphase spreads and works by preventing spindle fibers from pulling apart the duplicated chromosomes lined up in metaphase. Coopersmith was not used in this metaphase spreads protocol because it has been observed to alter the natural structure and organization of the chromosomes, which counteracts the initial purpose of revealing chromosome morphology. Therefore, this is no reflection of the SYTOX Deep Red stain itself. Instead, judging by the well-defined strands of fluorescent chromatin in the interface cells of SYTOX Deep Red compared to those in the DAPI stain, SYTOX Deep Red is observed to be optimal in its clarity and distinction of the double-stranded DNA. SYTOX Deep Red also has excellent photostability, reflected in the stained cell's high fluorescence. Although it was previously unknown how SYTOX Deep Red mechanistically bound to double-stranded DNA, it can be concluded that SYTOX Deep Red binds via hydrogen bonds to the major groove in the adenine-thymine region of double-stranded DNA, because all other modes by which a counterstain can bind are electrostatic interactions, minor groove hydrogen bonds, and intercalation, all of which would work similarly in DNA and either RNA or single-stranded DNA. This explains why SYTOX does not have high affinity for single-stranded DNA or RNA, a beneficial attribute for solely staining for chromosomes. In conclusion, the 1 to 4,000 dilution of SYTOX Deep Red is the most optimal, a significant discovery for future research using microscopy to understand how chromosome structures and organization lead to disease.